Hello, everyone, and welcome to today's live broadcast, Overcoming Hurdles in the Study of Rare Diseases, Innovative Strategies for Novel Therapy Development, presented by Jill Weimer. She is a Senior Director of Therapeutic Development and Associate Scientist in Children's Health Research Center at Sanford Research. I am Marjorie Torres of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom or center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Jill Weimer. I will now turn the presentation over to her.
Thanks, Marjorie, and I just want to start by thanking LabRoots for asking me to host and present as a part of this seminar series. What I'm going to talk about today is some unique approaches that my team has had and taken over the last couple of years in studying rare diseases. And before I jump into the meat of the presentation, I want to just give an overview of sort of the big picture approaches that my lab takes in studying cortical development and rare diseases. So many people and a lot of focus has been on in the last couple of years and sort of this bench to bedside approach. And so my lab is particularly interested in how the cortex develops in the mammalian brain. And so we ask a number of very basic questions about how different signaling molecules tell stem cells within the neurogenic niche to divide and migrate to their final resting place in the cortical plate. But while we're doing that, we're also looking at that interface of translational science. How do mutations in particular genes or disruption in some of these signaling pathways lead to neuropediatric diseases? And how can we use this knowledge gained from the basic science to develop effective treatments for these diseases? So we have projects in our lab that look at the proliferation of neuroprogenitors or neural stem cells or radial progenitors in the ventricular compartment. And so some of these projects look at small row GTPases or the regulator's guess in regulating the placement of these neuroprogenitors. We have projects in the lab that look at molecules that are important for neuronal polarity and the actual migration of these early excitatory projection neurons along the radioglial scaffold, in particular looking at a protein called MARX that we believe is important for early progenitor polarization, but once the cells actually reach the cortical plate, is important for the differentiation of these neurons and the extent of their axons and the ultimate pathfinding of those axons to their target in different parts of the cortex or lower subcortical structures. We have projects in the lab that look at the actual process of differentiation and long-term maintenance. It's really within this space that I'm going to focus a bulk of our talk today. So we look at factors that are important for regulating interaxonal trafficking, so the movement of organelles or cargo within the axon, and a number of the factors that we look at in this regard are involved in a rare lysosomal storage disorder or a family of lysosomal storage disorders called Batten's disease, and then also some implications in neurofibromatosis type 1. And then lastly, how do those factors actually go together to form the mature cerebral cortex? So focusing on that last box and sort of the differentiation and maintenance, we're looking at sort of how cargo moves within axons. And I think that often when we're learning biology, we learn that a cell is round, that all the organelles are located around the nucleus. So similar to what I show you in this cartoon, that you have this ER that's contiguous with the nuclear envelope, and positioned around that we have lysosomes and mitochondria. But neurons are very unique. They don't have this concentric placement of organelles because oftentimes the axons have to communicate over very long distances. And so those organelles that are usually confined to the cell body now have to be trafficked within the axon. And so we see movement along a microtubule railroad track, per se, of anywhere from synaptic vesicles to mitochondria. The smooth ER can flatten out into this space. And these cargo are transported along this microtubule by motor proteins. Kinesins are often moving things away from the cell body, but there's also return of this cargo to the cell body itself in order to allow for signaling back to the cell body or degradation of proteins, and this is usually done, this anterior grade transport, or sorry, retrograde transport is often done by motor proteins called dyneins. And so this movement of cargo is absolutely essential. We know that if we disrupt this, it can lead to a whole host of different disease states. The most common that we're all familiar with is the hyperphosphorylation of tau that we see in tauopathies or in Alzheimer's disease. Tau is one of these proteins that's responsible for binding to the microtubule network and stabilizing the microtubule network. And when you have hyperphosphorylation or disruption in tau, you lose the ability of that cargo to move cargo away from the cell body. And so knowing that this process is absolutely essential. And so what we're interested in the lab is understanding how various proteins are responsible for serving as sort of molecular tags or barcodes for the cargo that's being transported along this microtubule network. And so we centered around a protein called CRIMP2, and CRIMP2 is shown here in this smaller inset box of these organelles being trafficked along the axon. And CRIMP2 we kind of think of as the railroad car itself. So the kinesins, as I mentioned, or the dyneins would be kind of the train cars along this microtubule railroad track, and the CRIMP2 is serving as sort of the railroad cars. And just like we think about movement of traffic along railroad tracks, those cars can be loaded with a number of different cargo. And so I show here just the number of the known cargoes for CRIMP2, and I have many of those listed on the right-hand side. And today we're going to focus in on this blue represented cargo called CLN6. I will draw your attention to the fact that our lab does a lot of work on neurofibromatosis that I show here in pink, and it's a binding partner of CRIMP2. NF1, or neurofibromin, is the protein that's mutated in neurofibromatosis type 1. And so the sort of innovative approaches that we've actually taken to studying NF1 focus around development of novel animal models to study NF1. So like many diseases, when you mutate NF1 in mouse, the mice don't phenocopy the disease. And so many of the characteristics that we see in the human disease don't present in the mouse. So recently, in collaboration with Exemplar Genetics, a team at the University of Iowa and collaborators at the University of Arizona, we've developed a porcine or a pig model for neurofibromatosis. Unfortunately, I don't have time to tell you much about that model today, other than to say that it does show a lot of the human-like characteristics that are absent in the mouse, and presumably will provide the NF community with a much more accurate model of neurofibromatosis. So the remainder of today, we're going to focus on this interaction with CLN6. And what I hope to do is kind of take you through sort of our interest in the basic biology and what we think CLN6 is doing in neurons, but then also kind of tell you how we've used CLN6 as a central platform for the study of Batten disease, and come up with methods to accelerate the development of treatments once we have the appropriate tools in hand. So CLN6 binds to this CRIMP2, and up to this point, up to sort of when we started studying this protein a few years ago, what was known about CLN6 is that it's an ER-associated protein. And mutations in CLN6 can actually lead to two different forms of Batten disease. And I'll talk a little bit more about Batten disease here in a second. But really up to this point, folks had studied CLN6 in non-neuronal cells. But one of the things that's unique about Batten disease is that although these mutated proteins are expressed throughout the body, really the disease seems to be very specific to the central nervous system, particularly in the loss of neurons and activation of glia. So we took a different approach in saying, could there be something that's very unique about CLN6 and what it's doing in neurons as compared to cells within the rest of the body? And one of those things would be the transport of organelles or cargo down the axons. And so we kind of focus on that. Let me step back and just tell you about Batten disease, because it's oftentimes people haven't heard of this very rare disease. So Batten disease is also known as neuronal ceroid lipofuscinosis. And that name tells you a little bit about the disease in that in neurons and actually in other cells throughout the body, Batten disease patients accumulate a ceroid or lipofuscin. This is a lipopigment that accumulates within the lysosome. So this is considered a lysosomal storage disorder. And there are over 10 different variants of this disease that are caused by mutations in as many as 14 different genes. And really sort of the common element that links all the forms of Batten disease together is the accumulation of that ceroid within the cells, and then also a common set of phenotypes that we'll talk about here in a second. So overall, the incidence rate of Batten disease is 1 in 100,000 live births across these different variant forms. But collectively, that makes it the most common neurodegenerative pediatric disorder to date. It's incurable. There are treatments that treat some of the symptoms of the disorder, but ultimately it results in premature death. And just to give you sort of a look at what happens to a child with CLN6 Batten disease, so the variant infantile form of this disease, the kids are usually born fairly healthy. Around the age of 3 to 4, parents start to notice that they're having some language deficits, they're having some motor deficits, not keeping up in terms of their coordination and skills with their peers. And slowly they start to see a deterioration in their cognitive skills. So this is a regression. It's not a delay in onset. These kids are usually functioning completely normal compared to their peers. And over a period of a year, they really start to regress. Within a couple of years, they've lost almost all motor ability, they're wheelchair bound, they start to have very severe seizures, oftentimes they'll have visual deficits, and with the CLN6 Batten disease, these children usually don't live past their 11th or 12th birthday. So it's a horribly devastating disease with no treatments or cures. And so what our lab has focused on, again, as I said, is sort of the basic biology and then the translational. So just to give you a roadmap, we're going to spend the first portion kind of going through what we've done in the basic biology, and then we're going to switch over to more translational applications and really going back to this sort of innovative approach to accelerate the science. So the first question we started looking at is whether this interaction with CRIMP2 and CLN6 could mediate vesicular trafficking. Could it have this unique neuronal function that I alluded to earlier? And so the first thing that we did is start to look at how this complex actually comes together. And so I told you that these CRIMP2 molecules often dock to kinesin light chains. So we used yeast-2 hybrid a number of years ago to show that sort of the linker molecule to the microtubule network is this selection kinesin or kinesin light chain 4 that's able to bind to CLN6 and CRIMP2 and form what we call the CCK complex. And you can visualize this here in neuronal axons, where we're looking at, in the top panel, CLN6 GFP with CRIMP2, and you can see that a subpopulation of the CRIMP2 positive vesicles or puncta do co-localize with CLN6. Same in the case for the panel below that, where we're looking at localization of CLN6 and KLC4, and then with CRIMP2 and KLC4. And in the bottom panel, really, if you focus on that, there's a subpopulation of these cells, are these, cargo vesicles or puncta that are triple positive for all 3 of these molecules. So I just want to kind of pause here and say that as I mentioned before, CRIMP2 has a number of different interacting molecules that it binds to. So there was no assumption going into this that all of the CRIMP2 puncta would be CLN6 positive, but the same is the case for KLC4, where KLC4 also has a number of interaxonal cargo that it's able to bind to and transport besides CRIMP2 or CLN6. But there is a subpopulation that are positive. We're fortunate that there's a naturally occurring mouse model for CLN6 Batten disease. We spent quite a bit of time validating that this animal model does recapitulate the disease state that we see in CLN6 Batten disease. This includes retinal degeneration that starts around 3 months of age. There's motor coordination deficits that we can see between 3 to 6, depending on what the test is that we're doing. I show here that the autofluorescent storage material, we can see it present at 5 months. We pushed these limits, and we can see the autofluorescent storage material actually accumulating in these mice around 1 month of age. And then the glial activation, the astrocytic and microglial activation, we can see around 3 to 4 months of age. So really recapitulating in these mice much of what we see in the disease. And so we started to look at the neurons within the brain of these CLN6 mutant animals. So if we take out primary cortical neurons and put them into culture, and in this figure we're looking at on the top 1 day after culture, or on the bottom looking at 5 days after culture, what you can see hopefully and appreciate is that the CLN6 neurons seem to be delayed in their ability to break polarity and differentiate. And so on the left-hand side, you can see that very early on, at 1 day, that there's a process that's really kind of leading the pack at the top that's going to become the axon. And if you look at 5 days in culture, you can see clearly an axon that's delineated coming out of the top of that cell, where the dendrites are kind of around the cell body. In the CLN6, that seems to be delayed, and so in the 1 day and in the 5 days, you can see sort of this delay in the ability of these cells to polarize, and sort of this has been quantified in terms of the extent to the axon over time in the right-hand bar graphs. So these cells definitely have a delay in their maturation and ability to develop their axons. If we look at this in a little bit more detail using a shoal analysis, where essentially you draw concentric rings around a mature axon. In this case, we're focused really on the axons. I won't talk today, but there are some deficits also in the dendritic branching. But if we just focus on the axons that we've kind of traced in the little schematics on the left-hand side from wild type and mutant CLN6 neurons, what you see is that there's actually a delay or a deficit in the collateral branching within the axon of these neurons over time. These were done in DIV7 neurons. And so you can see if you follow just the trace for the shoal analysis, the orange is the wild type, and you can see that as it moves away from distance from the, or radius from the cell body, that it's throwing off a number of collaterals, and the CLN6 neurons are always at a deficit. They're developing, but they're just not developing at the complexity of the wild type neurons. So if you think about this in the context of the disease, if you had this delay in the complexity or sort of a inability to form the proper connections, your brain is going to be at a disadvantage from the start. So we can also look at the connections that these axons would be making. So axons connect onto dendrites, typically at dendritic spines. What we're showing here is electron micrographs of Golgi-impregnated dendritic spines. And you can see these hair-like follicles, these dendritic spines that protrude. Here, just we're focused on the primary primary dendrite coming out of the top of the neuron. And if you count those dendritic spines, you'll see that there is a significant reduction in the CLN6 mutant animals, suggesting that they're making fewer synapses, likely because of the deficits in those axonal collaterals and axonal connections that are forming. So there is a deficit in the cortical neuron branching. If you look at a deficit in axon or axon number, ultimately that should lead to a deficit in fiber tract formation. And so what we did was perform electron microscopy in coronal cross sections across the corpus callosum and look at sort of the number of axons that are actually crossing at the midline. We did this at 1 month and at 10 months of age. And at 1 month, there seemed to be okay. They seem to be able to get the axons there. Interestingly, there is a deficit in the myelination or the myelin compaction around these neurons, even as early as 1 month. But if you look at 10 months, there's a definite reduction in the number of axons that are crossing at the midline, and in that number, that myelin compaction is maintained. So they're not maintaining these axonal fibers across the midline as they would in the wild-type animal. So I apologize, there's supposed to be a couple of video clips here, but essentially what we wanted to do next is look into the actual trafficking of these cargo vesicles. And so we can use a live dye called ER tracker with the assumption that CLN6 has been associated with the ER. Now, in the axon itself, there's two things that happen with the ER. There are small vesicles that pinch off at the ER and are trafficked very rapidly along the axons, but they're also, the smooth ER also flattens itself and is contiguous within the axon itself. And in these live videos, and I'll show you fixed frames if we move forward here, what you can see is that above that contiguous smooth ER, there are these very distinct, rapidly moving ER tracker positive puncta that move within the axon. These, if the video had worked, I can show you that these puncta actually pause at areas that are presumably synaptic connections. They might pick up a signal, and then you see them quickly move out of that space and continue down the axon. So if we take still frames of these movies, as I'm showing here, we can quantify the number of these ER positive or ER tracker positive vesicles in wild type compared to CLN6 mutant animals. And what we see is that there's about half the number of these ER positive vesicles that are moving in the CLN6 mutant axons, suggesting that that impairment in the CCK complex is precluding the ability of these vesicles to move correctly. So just kind of thinking about how this disruption might occur, we started to do a number of biochemistry experiments to look at whether loss of CLN6 is actually interfering with CRIMP2 or the kinesin proteins. So remember that I told you that CRIMP2 interacts with a whole host of different proteins. And if we start to look at some of those, what we see is quite interesting. So if we immunoblot for the kinesins, what we see is a significant elevation of KLC4 in the axons, or in the brain of CLN6 mutant animals, but we see a reduction in some of those other interacting proteins like NF1 and SARA1, which is a protein expressed at the leading edge of the axons. So what this has suggested to us is that the loss of CLN6 does interfere with the repertoire of proteins that CRIMP2 would normally be interacting with through these IP experiments. And so suggesting that when you lose CLN6, CRIMP2 is kind of binding tightly to the microtubule network and the kinesin proteins and losing its ability to interact with some of these other forward-facing cargo proteins like NF1 and SAR1. So that's sort of been our working hypothesis, that you have, as I show here in this cartoon, you have CLN6 that's transmembrane, which I failed to mention, transmembrane to these ER proteins. And we believe it's sort of serving as a molecular barcode on these ER-derived vesicles to bind to CRIMP and the kinesin proteins. But what is this cargo? What are these ER vesicles? We're taking a number of both unbiased and more targeted approaches to figure out what this cargo is. So one of the ways that we're looking at this is to use a technique called BioID. This is a technology where a promiscuous biotin ligase is fused to our CLN6. In this case, what we're doing is put in on that C terminus, or that yellow squiggly, the end of that yellow squiggly CLN6 protein, so that what it would be doing is tagging any proteins that are luminal to these ER tracker positive vesicles, so that we'd be able to tag cargo and look at that protein localization. in order to do this, we're packaging our CLN6 BRA BioID protein into AAV9, which gives very specific neuronal localization targeting through intracerebral injections. We'll then isolate vesicles or synaptosomal preps and do mass spec to see what targets are biotinylated in these proteins to identify interacting proteins or things that might be in these vesicles. We're also taking a more candidate approach, and I apologize, these bar graphs come up a little bit small, but what we've been doing is looking at proteins whose mutation lead to a similar phenotype. So in the 14 different types or genetic types of Batten disease, 3 of them are caused by mutations in lysosomal enzymes. PPT leads to infantile Batten disease. Mutations in TPP1 or CLN2 are the late infantile, and then mutations in cathepsin D lead to a fatal form of Batten disease. And so we've looked at the enzyme level of each of these proteins or its enzyme activity through a cleavage assay, tagging assay. And so essentially on the left-hand side in these graphs, we have enzyme activity assays from whole brain for PPT, TPP, and cathepsin D. And then on the right-hand side, we have enzyme activity assays for just synaptosomal preps. So although the proteins don't change much in terms of their enzyme activity in the whole brain, what you can hopefully appreciate is that in all cases, there's a marked decrease in the ability of these enzymes to act within the synaptosomal preps, which would presumably lead one to speculate that possibly these proteins aren't making it or being targeted correctly to the synapse itself. And so one possibility is that CLN6 is responsible for trafficking these particular enzymes to the nerve terminal, which would give a similar disease phenotype. So right now what we're doing is going back and looking by super resolution microscopy to see whether we can see co-localization of these enzymes in CLN6 positive vesicles within the axon. So stay tuned for that portion of the story. So just to summarize the basic science piece, what we've shown is that CLN6 forms this CCK complex with CRIMP2 and KLC4, and disruption in CLN6 can lead to a phenotype that's very similar to what we see with a loss of function of CRIMP2 and this failure to properly specify and maintain axons. So now kind of taking an approach where we know a little bit about the basic biology, could we actually target therapeutic complexes to this CRIMP2 associated CRIMP2 associated complex as a potential treatment for Batten disease? So we did this. We wanted to select a target that would do a number of different things, that it would help promote neuronal survival. It might decrease some of the glial activation that I told you was associated with Batten disease, that it might increase the neurite stability so we could get those axons branching and connecting correctly, that it might correct some of that ER tracker vesicular trafficking, and it might increase CRIMP2's ability to interface with some of those other proteins like NF1 or SAR1 and loosen its affiliation with the microtubule network and the kinesins. So there's a number of compounds that have been shown to target CRIMP2. I list those here, and they're reviewed in a review article that we put together with Raj Khanna a number of years ago in Future Neurology, if anybody wants to check those out. But for the purposes of our talk today, we're going to focus on this compound here called lanthiamine ketamine ester. This is a small molecule that binds to CRIMP2 and has been screened as a potential therapeutic in a number of neurodegenerative diseases, including ALS and mouse models of Alzheimer's disease. And so the other thing that makes LK or made LK particularly attractive to us is that it's a cell-permeating, brain-penetrating, orally bioavailable compound. So oftentimes when we're thinking about therapies for the brain, we have to know whether they'll actually readily cross the blood-brain barrier or we'll have to deliver them systemically to the central nervous system. The great thing about LKE is that we can just compound it into the mouse chow more than sufficient amounts are able to reach those primary neurons. Work from Ken Hensley's lab has shown that when you throw this compound, in this case in the A and B, onto neurons from the dorsal root ganglia, what you can see is that it really enhances their ability to send out neurites, and that's quantified in C. Interestingly, this compound too, from Ken's lab, it seems to also have some neuroimmune modulation effects. So in the E and F, looking at sort of the response in microglia or microglia cell line to the release of nitric oxide and sort of the ability of these cells to respond. So this really, for us, seemed like a very appealing molecule, because if we could increase collateral or branching, and then also dampen down some of the neuroimmune responses. But one of the other things that was particularly interesting or attractive to us about LKE is that it seems to have a counteractive effect to what we're seeing in the disease state. So recall that I told you that in CLN6 mutant brains, there seems to be an increased binding to kinesin light chain in the microtubule network and decreased binding to proteins, the sort of cargo CRIMP2 proteins like NF1. When you add LK, it actually does the opposite. So it binds less to the microtubule network and increases affinity for proteins like NF1 that I show here in these IPs. So this really told us that it might be able to counteract some of those defects that we're seeing at even the biochemical level in the NF1 CLN6 mutant animals. So we started a number of studies, both looking in vitro and in vivo at how this compound would work. And so I'm going to show you first just the in vitro studies. So the left two panels are those that I showed you before, where we were looking at day 1 and day 5 in culture of primary cortical neurons. And then in the right-hand side, what we have is those same mutant neurons, but now with the addition of LKE. And what hopefully you can see is that the bar graphs here are just comparing the mutant to the mutant with drug. So left-hand side is the CLN6 neurons, and then the light blue, Carolina blue bars are the mutant cells with the LKE. And what you can hopefully appreciate is that in all situations, the addition of LKE increases the initial break in polarity deficit that we see at day 1, and the continued maturation of those axons out at day 5. So LKE in this case is able to rescue that branching. If we look at the Scholl analysis, again, on top, the light blue is with LKE, the dark blue is what we had shown before with in just the mutant neurons. And again, the addition of the LKE is really getting these Scholl analyses of the collateral branching back up to wild type levels. So at in the cell culture level, it's able to correct the axonal specification, but what about the interaxonal trafficking defect that we saw with the ER tracker? So if we look now with the addition of LKE, it's able to partially restore the number of these ER tracker positive vesicles in the CLN6 mutant neurons. Now, I think that probably part of this is due to the fact that we're not completely restoring CLN6, so we're probably never going to be able to get the CLN6 positive vesicles or cargo themselves moving. But what we're hopefully doing here is actually correcting some of those other cargo that are decreased. So some of the maybe NF1 or the SAR1, some of those other CRIMP2 binding cargoes that are moving within the axons and partially rescuing the phenotype. So the next thing we wanted to do is look at, is this correcting any of the phenotypes in the animals themselves. So we dose the animals with LKE starting at weaning, and what we see in the top panel is the survival of these animals. So the CLN6 animals start to take a turn for the worse around 8 to 10 months, and all of the CLN6 animals are dead by about 12 to 14 months of age. And there seems to be this slow progressive loss of animals. The addition of LKE kind of delays the initial death, but ultimately the animals do succumb to the disease around the same age, around 12 to 14 months of age. So it's not, it seems to be delaying the early phase or the early onset of the disease, but ultimately long term they're succumbing to the disease. The next panel on B, we're looking at some visual processing. So we use a system called the visual cliff that I'm showing here. So the animals are placed in this chamber that has a plexiglass bottom, and typically the wild type mice will walk right up to the edge of that cliff, and they'll be a little bit nervous about crossing over because they think they're going to fall off the edge of the cliff. The CLN6 animals that are shown in the middle gray bar have a hard time distinguishing that cliff, and so they'll move back and forth across that boundary space much more readily. But the animals that were treated with LKE seem to be on par with the wild type animals and in their ability to cross over that space. And when we start to look at some of the other parameters, whether this is in C in a radial arm maze, looking at learning and memory in late stages of the disease, you see a little bit of correction with the LKE. In motor performance or open field, we don't see quite as much of an improvement with the LKE, although it seems to be somewhere an intermediate phenotype as shown in D in open field. And then in the bottom in rotor rod testing to look at some of those motor parameters. Unfortunately, the LKE doesn't seem to rescue any of the motor parameters, and the mutant animals and those with drugs seem to be in line with one another, whereas the wild type is up above and maintains some of that motor performance and coordination that you would associate with rotor rod. I just show those behavior tests there. So what does it look like at the pathological level? So if we start to get into some of the brain phenotypes. So first I'm showing you CD68 immunohistochemistry, and we focus in our Batten disease studies in two different brain regions. The first on the left is a cortical region, S1BF. So this is the somatosensory barrel field cortex of the mouse, and this is from work of John Cooper and some of our early characterization of the mice. Sort of seems to be the targeted subcortical region that is most severely affected. And then on the right-hand side, where that somatosensory cortex connects to in the thalamus, one of the subnuclei, the VPN, VPL of the thalamus, is also an area that seems to be very targeted. And interestingly, the addition of LKE actually seems to exacerbate the microglial phenotype. So the lower right-hand panel is the mutant animals with LKE compared to the untreated mutants in C. And in all instances, there seems to be this significant elevation in microglial activation. And I will say that even in the case of the cortex, in the wild type animals, just simply adding LKE can make these microglia hyper-responsive. Now, we don't know whether the microglia are simply responding to the disease state or whether they're actually contributing to the pathogenesis of the disease. We use this as a readout. So I would caution us that we couldn't and shouldn't make a single call on whether a drug is effective on whether we're seeing changes, because maybe they're actually, this drug is just making the brain a little bit more hyper-aware of what's going on. But if we look at astrocytic activation, so in this case with GFAP reactivity, we see the same thing, that there's, if you compare wild type to untreated mutants in the top left to the bottom left of these panels, you can see a significant elevation in GFAP reactivity, but this is very, very heightened with the addition of LKE, suggesting that LKE is making these, both microglia and astrocytes, just hyper-reactive. If we look at the storage material, so recall that I told you that the hallmark of this disease is that they accumulate storage material in the lysosomes. The same thing is true, that you really don't see any preservation or any protection with the addition of LKE in the brain, either in the cortex or the thalamus. And in this case, what we're looking at is immunohistochemistry for one of the constituents of this disease, the subunit C, mitochondrial subunit C ATP synthase, which we know accumulates in these cells. So sort of the take-home message is that the LKE isn't working. We have looked at a number of other CRIMP2 targeting compounds, and it seems to be the case, too, that although you can get some correction at the cellular level and maybe modest phenotypic changes at the pathological level, we're not really seeing much in terms of recovery if we're targeting CRIMP2 as a therapy. So just to summarize this portion of the talk, we've looked at these LKE compounds, or these CRIMP2 targeting compounds, to see whether they might have some potential therapeutic benefits. But the treatment of Batten's disease is likely going to require a combinatorial approach. So I don't want us to say that we're going to completely rule out CRIMP2 targeting compounds, because as I showed you, they do have a slight phenotypic improvement, but we really think they're going to have to be offered in combination with something else. So that caused us to kind of rethink our strategy in the lab about 2 years ago. And this kind of came in combination with working with a CLN6 foundation called the Charlotte and Gwyneth Gray Foundation. And really sort of this was a new way of looking at team science. So our lab in this sort of large program grant sits at the center as the CLN6 Batten disease, but we brought in experts from all these different therapeutic modalities, looking at antisense oligos, or using IPS for disease modeling, iPSCs for disease modeling, using gene therapy as a potential treatment, and looking at a whole host of different molecules. And we wanted to do a couple of things. We wanted to come up with a system where scientists really were at the table for the same common end goal, and not in the program for their individual science. So sort of the innovation in this is that all the members of the team really kind of check their egos at the door, and are taking this holistic approach and team science approach to the treatment of Batten disease and coming up with therapies. But the other thing that we did was sit down and say, how can we accelerate drugs through the pipeline? So many of you are familiar with schematics similar to this, showing sort of the timeline that it takes to get drugs through the therapeutic pipeline. And so it might take 4 to 6 years to screen, to do, say, a high throughput screen, where you'd be looking at, 5 to 10,000 different compounds. Through those high throughput screens, you might narrow your drug targets down to a couple hundred. You would then go back and start doing some preclinical testing, whether it would be on human cell lines or in animal models. And only after about 5 to 7 years have you narrowed these down. Oftentimes you narrow it down like we did in the case of LKE, spend a few years studying that molecule in long-term animal studies, only to discover that it doesn't work. And so now you're back at the drawing board before you would even go into a clinical trial, which in many cases can take 6 to 7 years to actually move a drug to market. And so we wanted to come up with an approach where we could actually move this faster. So one way in terms of the molecules is that we've developed a very rapid, high content screening platform for moving drugs through in vitro studies, and then short-term in vivo before we would even take them into long-term. And so at each step of this process, we're eliminating drug compounds. And for the Batten disease studies, we're really focused on things that are already approved FDA compounds or things that are in the approval pipeline. We then screen those in either mouse lines, patient IPS lines, or patient fibroblast lines, and then go into sort of short-term pathology to make sure that it's working before we would ever go into long-term drug studies. And by doing this, we're able to eliminate a lot of compounds, but essentially the way that this high content screening works is you can look at a number of different parameters and screen a number of different drugs. So this is just a sampling of the drugs that we've been looking at over the last couple of years. But essentially you can look at many, many different parameters. So in this case, we're looking at autophagy using a cyto-ID. We're looking at lysosomal pH using lysosensor. We can look at mitochondrial function in these cells. We can look at cell rocks to give an indication of reactive oxygen species. For some reason I have autophagy twice. But you can also use high content screening to look at antibody-based or ICC in these patient cell lines, looking at things like the placement of the lysosomes. You can look at apoptosis, you can look at cell proliferation, and then do other assays on top of this, looking at things like mitochondrial function in a seahorse assay, or looking at cell stress. So it really allows us to look at a number of different parameters. We don't confine ourselves just to patient cell lines like fibroblasts. We can also look at neurons and look at the complexity of these neurons in terms of axonal specification and outgrowth in response to drugs, and look at a number of different parameters, like look at synapse numbers and look at unique cell populations. there's a number of factors that I think that we often ignore when we're doing high throughput or high content screening or even in vitro assays, that I think that these high content assays really allow us to dissect. So I'm just going to list a number of these things that we're thinking about and looking at now, whether there's genotype or phenotype relationships. So do different mutations, even in the same gene, lead to different types of phenotypic changes? Are there sex differences? So in Batten disease, I can tell you that we often do see male and female differences, but at the level of the animal, scientists often disregard that, particularly when they're doing cell culture. Are there differences in disease state and progression? So with those different genetic mutations, can we see changes in the disease status and progression over time? I'm losing my mouse. The relevance of the ease of the assay itself. So the high content can give us very reliable, reproducible results that we can rapidly move through a number of compounds fairly quickly. And then it also allows us to ask questions about things that are actual disease indicators versus functional attributes of the disease. And then we can use these high content assays to start layering drugs together much more rapidly. So instead of doing drug screening sequentially, we can actually start to put these together. So just in the last few minutes, I want to tell you about sort of one of our big success stories in coming up with this more rapid model. And this was really, when we were looking at all of those different modalities, we knew that gene therapy probably had the highest likelihood of getting us to the finish line the fastest, while we were continuing to look at a number of those other molecules. So we partnered with a team at Nationwide Children's Hospital that had been very successful in developing an AAV9 treatment for spinal muscular atrophy. They would have been able to take this AAV9 SMA through clinical trials, and we're seeing very promising results in the kids. So what we decided to do is actually piggyback onto their successes. So we packaged the CLN6 human gene into their AAV9 plasmid that has a chicken beta-actin promoter, so it's going to give us very robust expression throughout the central nervous system. There was some indication that overexpression of some of the NCL proteins might be cytotoxic to the cells. So we started off by just expressing this plasmid in non-neuronal cells, looking to see if it interfered with cell survival. You can see here that at the RNA and the protein level in these cells, they look completely healthy and they're able to produce large amounts of CLN6. If we electroporate this plasmid into the brain of an embryonic day 15-old mouse and allow that animal to continue to develop out to postnatal day 7, what you can see when we co-electroporate GFP with the human CLN6 plasmid is that we get very nice expression of CLN6 in neurons in the cortical plate. It extends into the processes similar to what we had seen in our culture model. So just looking here in the red, comparing that to the GFP co-labeled cells. What you can see is that there appears to be no toxic effect. So overexpression of these proteins, or this protein in particular, doesn't really seem to have any toxicity in the animals. And so we moved forward with injecting this into the mice for an efficacy study. The tropism of AAV9 changes slightly between mouse and non-human primates and humans. So in these particular studies, we give a single intracerebral injection of the AAV9, starting at, we give that injection at postnatal day 1, and then take these animals out as long as they will live. And so through that intracranial injection, we get nice increase in RNA expression of CLN6 throughout the central nervous system, all the way down the spinal cord. So this virus is able to spread very nicely. If we look at in situ hybridization for CLN6, in the bottom row, you're looking at a probe specific for CLN6, and you can see nice spread of this virus or RNA expression of this from this virus throughout different regions of the central nervous system. And here is really kind of the most telling slide. So this is using, in the bottom, an antibody that is specific to the human CLN6, and this is in CLN6 mutant mice compared to a PBS-injected control animal on the top. You can see really nice, robust expression of CLN6 throughout the central nervous system and all the brain regions that we looked at from that single ICV injection. So what we've done over time is look at sort of the recovery of the phenotype, both pathological and behaviorally. So here I'm just showing you autofluorescent storage material. The left-hand column is wild type. The middle column is CLN6 animals injected with PBS. And then the far right column is CLN6 mutant animals injected with the gene therapy in 2 different brain regions, again, somatosensory cortex and VPN, VPL of the thalamus at 3 different time points. So on the top, we're looking at 2 months of age, middle panels is 6 months, and then the bottom 2 panels is actually all the way out for the wild type in the gene therapy treated animals at 18 months of age. The middle panel is a comparable image from a 14-month animal. At 14 months of age is when all of our CLN6 PBS treated animals are dead, but our mutant animals treated with gene therapy continue to live past that. If we look at the subunit C accumulation at these same time points, the gene therapy is able to stave off that storage material accumulation all the way out to 18 months of age. So it's really preventing the onset of this disease very nicely. You can see that in terms of astrocytic activation here with GFAP positive astrocytes, and the same with the microglia. And I will tell you that the microglia in the middle panel, this is again when these animals are moribund, they're on their way out, and so these microglia themselves are starting to look really sick. They're not just hyper reactive. So they're looking really ill. But in the CLN6 that are treated with gene therapy, you see a little bit of microglial activation, but it is nothing compared to the untreated mutant animals. So behaviorally, what is happening to these animals? I'm just showing you a number of different assays. So on the top left, this is rotarod. The mutant animals that are untreated are at the bottom, and our gene therapy treated animals are indistinguishable from the wild type. So the light blue and the black or the gray charcoal bars are staying together. The same with, if you look on the middle and left-hand side, we're looking at Morris water maze as a measure of learning and memory. The mutant animals are have an inability as time goes on to find a platform for Morris water maze, but the mutant animals that are treated with gene therapy are really on par with the wild type animals. Probably at the later stages, they're starting to have a little bit of declining. You can see a slight difference, but really it's substantially better than what we see for the mutant animals. Their body weight is maintained, they're healthy, they're happy. In those later stages at 12 and 18 months of age, just even comparing the wild type to the mutant animal, or the, gene therapy treated mutants to the wild type animals, they're really indistinguishable. They're grooming, they're walking, they're behaving, they're very similar to the mutant animals that you can see from these clasping scores. But really this is kind of the big take-home message, is that the mutant animals are that solid blue bar that drops off. These are the untreated animals, and as I said, they're dead by 12 to 14 months of age. But the survival of the gene therapy treated animals in the light blue bars is really on par, and these animals are surviving out to 24 months of age. So probably one of the best long-term treatment approaches that we've seen. So the next step, of course, would be a clinical trial. So what we were able to do in this particular study is that in collaboration with the team at Nationwide, we stacked all of those things that we would normally do in a very linear process on top of one another. So instead of doing efficacy studies, waiting for those results, and then doing toxicology studies, waiting for those results, and then injecting the virus into non-human primates, essentially building on the success that Nationwide had, we put those whole processes on top of each other. We were simultaneously injecting non-human primates. At the same time, we were collecting efficacy data, which allowed us to go to the FDA within 8 months of starting the study and receive IND approval to start a clinical trial. That clinical trial is ongoing at Nationwide Hospital. They've completed the early phases of this, which enrolled 6 patients, and now they're looking to expand inclusion in that clinical trial. That's a phase 1, phase 2 safety trial that's currently ongoing. So stay tuned to see whether this is working, but it really sort of that accelerated pipeline drawing on previous expertise and knowledge has really allowed us to move this pipeline along a lot faster. So just to summarize, I kind of go back to this slide. We have a number of ongoing studies that are continuing to look at other treatment modalities, now kind of taking the approach that if the gene therapy continues to be successful, what we'll ultimately need to be able to do is offer potentially other drugs in combination with gene therapy. So instead of even screening these alone or maybe a couple of drugs at a time, we also conduct these studies in combination with gene therapy, because there's a lot of unknowns about gene therapy still. We don't know how long the virus will last. Hopefully it will last the lifetime of these children, but we have to have something else in our arsenal that's ready to go should the gene therapy start to wane over time. So I'll just wrap up here. I'll show my research team. Really a lot of the gene therapy work that I highlighted was led by Jacob Kane, a postdoc in the lab, and 2 technicians, Katie White and Derek Tim. The basic biology work was spearheaded by another postdoc in the lab, Sung-Yoon Ko, and one of the graduate students, John Brudvik, that's helping her finish up those studies. The gray foundation team I've shown here in the middle, these folks have been absolutely instrumental in the work that we've been able to move forward and that we continue to do. Our CRIM2 collaborators, and lastly, I just want to recognize our support that we receive both from the NIH and from a number of Batten disease foundations and NF1 foundations. Thank you very much.
Thank you, Dr. Jill Weimer, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window and type your questions into the box that appears on your screen, and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what attracted you to rare disease research?
I think there's a number of things that I find attractive about rare disease research. One of them is sort of the limited focus that's been paid to these diseases over time. So even though every single rare disease is individually rare, there's over 7,000 rare diseases. And oftentimes, you know, over a decade ago, a lot of the focus was really on the more prevalent diseases like Alzheimer's or Parkinson's disease. But a lot of scientists are realizing that we can glean a lot of information from rare diseases, both on the basic biology and therapeutic development side of things. So oftentimes we look at this as if we can get a drug approved and potentially to the market for a rare disease, that same technology or even possibly even that same drug might have applications for these more prevalent diseases like Alzheimer's or Parkinson's or other neurodegenerative diseases. So I think that part of it is sort of the compassion for the patients, that there's nobody or very few people that are fighting their fight for them. But two, on the flip side, from the scientific side, that we could have a larger impact by developing tools and technologies and a way of thinking in rare diseases that could be applied to other disorders.
Thank you for that answer. Our next question is, what links all the forms of Batten diseases together? Do the proteins involve all functions in a common pathway?
You would think that because there's such a similar phenotype, that there might be some cellular mechanism that is similar across all forms of Batten disease. But so far, we haven't been able to discover any common feature. In fact, even though these diseases are classified as lysosomal storage disorders, they're not even all lysosomal proteins. So for instance, CLN6 only spends a little bit of time in the lysosome. Most of its time is spent in the ER. And so far, we don't think that there is a common feature. I mentioned PPT, TPP1, and cathepsin D. Those are all lysosomal enzymes, so those at least are linked together in sort of a common cellular function. We do believe that possibly CLN6, CLN3, and CLN8 might be linked together in a common role in trafficking. So there's quite a bit of evidence that each of those proteins interacts with some of the microtubule binding proteins like kinesins, dyneins, and myosins. So they might also be clustered together into a common pathway. But you know, really, other than those, there's not a real common functional feature. What is common is that they all result in this common phenotype, the storage material accumulation, and a common pathology.
Thank you. Okay, it looks like we have time for one more question. Our last question is, your studies use ICV to deliver gene therapy to a brain. Do you think this is the best delivery method, or could other methods be better?
So there's actually a number of investigators, even in the Batten disease space, that are looking at IV infusion of gene therapy. And part of the rationale for doing that is that in some of the forms of Batten disease, there does seem to be long-term, other more systemic changes in the body. So the idea is, could they actually deliver systemically and correct both the brain and the other non-central nervous system changes that they're seeing. In CLN6 in the animals, we haven't really seen a lot of non-CNS pathologies. We've actually looked at the mice out to probably about 18 months of age, and really, or sorry, not 18, out to 1 year of age. And we're not really seeing any non-CNS changes that might be contributing to their death. So I think in the case of CLN6, targeting to the CNS is the way to go. This is the organ that is most severely affected. It's really, what's going to keep these kids alive. And then also, just like with drugs, the ability of the virus to cross the blood-brain barrier, although it does it a little bit, it doesn't do it super efficiently. So if we give an ICV injection directly into the brain, some of the virus does get out of the brain to target those other organs, but the same is true if you give the IV injection. You're limiting the amount that can actually go into the brain. So in our cases, ICV does seem to be the way to go, really so that we can correct those neuronal phenotypes or those central nervous system phenotypes. But only time will tell if we're going to uncover other non-CNS pathologies that ultimately these kiddos are being affected by.
I would like to once again thank Dr. Jill Weimer for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Goodbye.